Welcome back to The Explainer. Today we are unpacking something that's basically aiming to tame the often chaotic world of computational chemistry. We're looking at BioDockify, a platform that essentially serves as the AI operating system for modern pharmaceutical research.
Okay, let's dive into this. To understand exactly why a tool like BioDockify is making such massive waves right now, we really got to look at the numbers. The global bioinformatics services market is exploding. It's growing at a 14.5% compound annual growth rate. That puts it on track to hit a staggering $12.39 billion by 2034. Why? Well, because the pressure to lower costs and dramatically compress the timelines for drug discovery is literally off the charts. But to actually achieve that, researchers desperately need better, faster, and much more integrated tools.
So here's our logical workflow journey for today. We'll start with the fragmented pharma problem, explore the AI operating system, look at literature and research AI, docking and molecular dynamics, analysis and publishing, and finally wrap up with the future of discovery. It's a massive 16-module ecosystem, so let's get right into it. All right, kicking things off with Section 1, the fragmented pharma problem, or really, the cost of disconnection. Look, if you've spent even 5 minutes in computational drug discovery, you already know the absolute frustration here. The old way is just a mess of context switching. You've got one clunky, disconnected tool for your literature review, a totally separate one for molecular docking, some painful local cluster setup for molecular dynamics, and then, another piece of software just to write the paper. Plus, you're usually locked into whatever vendor you chose early on. BioDockify completely flips this script. It promises a unified AI operating system. It's entirely cloud native, meaning zero installation required. You literally just open your browser and go. And as we'll see, it is proudly provider agnostic. You actually spend your time doing science, not playing IT support.
Which brings us perfectly to Section 2, the AI operating system, a unified ecosystem.
And this brilliantly illustrates the sheer scale we're talking about here. The BioDockify ecosystem isn't just some standalone application. It encompasses 16 fully integrated modules that cover the complete computational chemistry workflow. We're talking everything from AI-driven literature intelligence to molecular modeling, docking, advanced molecular dynamics simulations, and even collaborative research sharing. All of these core components seamlessly talk to each other. Imagine going from a rough research hypothesis straight through to a docking simulation without ever leaving the platform.
Now, I want to highlight something absolutely crucial. Their core philosophy is that researchers should be AI-assisted rather than AI-dependent. They aren't just throwing a black box AI at you to make complex scientific decisions. Instead, you maintain total 100% transparent control over every single experimental parameter. The AI is simply there to act as a massive accelerator, organizing your data and handling the tedious, repetitive stuff. But the actual science, the critical decision-making, that stays firmly in your hands.
And a huge part of keeping you in control comes down to their provider-agnostic AI layer. BioDockify features this brain interface that lets you pick whatever AI model fits your specific needs. Want to plug in your own API keys? Go for it. Want to leverage NVIDIA NIM integration for cutting-edge models like MALMIM, or maybe use OpenRouter? Easy. You can even hook up local open-source models if you're dealing with strict data privacy requirements. There is absolutely no vendor lock-in here whatsoever. You choose the brain that powers your workspace.
Let's transition to the first real phase of the journey. Section 3, Literature and Research AI, Automating Discovery.
We all know any serious project starts with a deep dive into the literature. BioDockify's deep research module doesn't just ping 1 or 2 sites. It hits up to 10 databases simultaneously. We're talking instant parallel queries across PubMed, Semantic Scholar, Crossref, OpenAlex, plus major preprint servers like Archive and BioDockify. The AI instantly synthesizes mountains of literature, helping you spot knowledge gaps and generate hypotheses in a fraction of the time it would take to manually scroll through hundreds of PDFs.
Now, what's really interesting about this setup is how BioDockify actually delegates these complex workflows. They use 4 specialized AI subagents that basically act like your own personal collaborative assistants. You've got the researcher agent digging through literature synthesis and patent APIs, the biostatistician chewing through clinical trial data and PK/PD modeling, the writer drafting your papers, and even a hacker agent handling custom code execution and web scraping. It's honestly like having a fully staffed postdoc team available 24/7 right inside your browser. Moving right along to the core computational stuff. Section 4, Docking and Molecular Dynamics, the heavy lifting.
BioDockify takes the notoriously clunky traditional docking process and boils it down to a super straightforward three-step workflow. Step 1, you literally just drag and drop your receptor and ligand files. It accepts formats like PDB, SDF, or even raw SMILE strings, and it auto-prepares the receptors for you, completely wiping out the nightmare of manual file conversions. Step 2 is consensus scoring, which fires up multiple docking engines simultaneously. And step 3 is the results dashboard, giving you this gorgeous interactive 3D viewer with a complete energy breakdown, all in a matter of minutes. Let's zoom in on that consensus scoring for a second, because this is a fascinating approach to binding predictions. It runs a powerful three-engine process. First, it takes Autodoc Vina for traditional physics-based scoring. Then it pairs that with GINA, which uses a convolutional neural network for deep learning pose assessment, and adds SMINA to the mix. Finally, it refines the top hits using MMMGBSA for highly accurate free energy calculations. By fusing old-school molecular mechanics with modern deep learning, the reliability of your binding predictions just skyrockets. Of course, once you nail a great docking pose, you need to validate its stability over time. Enter the MD Lite module for GPU-accelerated molecular dynamics. You can seamlessly run 20 to 100 nanosecond trajectories using OpenMM and GROMACS entirely in the cloud. You just kick off a 24 to 48-hour background run and let the system handle the auto-resuming from checkpoints. It 100% eliminates the need for you to maintain or pay for some massive local computing cluster. Once those simulations wrap up, we move to Section 5, Analysis and Publishing, from data to paper. So the crucial point here is that you can finally ditch separate expensive software like SPSS or Jamovi to do your biostatistics. BioDockify has a native built-in statistics engine that gives you Jamovi-level parity right out of the box. We're talking over 20 specific test types, a full Bayesian suite for calculating Bayes factors, and this incredible auto-analyze function. You literally click a button and it spits out a beautifully formatted PDF report on their letterhead, complete with methodology and interpretation sections, all built with FDA and EMA compliance in mind. And when it's finally time to actually write the manuscript, the academic writer module takes over. Here's a wild fact for you. Researchers routinely waste weeks, literally weeks, of their lives just reformatting references and tweaking margins to appease the arbitrary guidelines of different journals. The academic writer eliminates that friction entirely. It gives you an 8-tab writing assistant to draft everything from your thesis to grant proposals, plus a journal finder that queries over 36,000 verified scientific journals to find the perfect home for your research, automating all that tedious formatting for you. Finally, let's bring all of this together in Section 6, the future of discovery, your AI workspace. Let's move to and see how this builds a persistent, ever-growing knowledge base. The BioDockify AI memory system relies on a PG vector-powered retrieval augmented generation, or RAG environment. So what does that actually mean for you? It means the software simply never forgets. It indexes every single paper you upload, every protein target, every chemical compound, and every chat log you have with the AI. Whether you are halfway through a 3-year PhD or wrapping up a decade-long commercial project, your workspace scales right alongside you, allowing for instant semantic search across your entire history of scientific thought. Which brings us to the ultimate point of this explainer. The real goal of BioDockify isn't just to throw cool features at a wall. It's to remove the friction. Think about it. If your software seamlessly handled all the literature gathering, the data formatting, the agonizing cluster configurations, and the statistical reporting, what new medicines could your mind discover with all that freed up cognitive space? It's an absolutely fascinating question, and one that makes the future of pharmaceutical research look incredibly exciting. Thanks for joining me on this deep dive, and as always, keep learning.